Nuclear magnetic resonance works by flipping around the magnetic moment of atoms in your sample. And by manipulating that magnetic spin, we can tell what other things are around that nuclei. So think of it like little magnetic fields. If I start that magnet spinning around, it senses the other magnet spinning around nearby, and the way in which they feel each other, they interact in various ways that we can detect.
We've done a number of different metabolomic study. We've done brain extracts, serum extract, fecal extracts. Anything you can get into solution, we can sort of look at.
It takes very little processing, so that's one of the nice features of NMR. You just take the sample that you've gotten from the patient and just add a little bit of buffer solution to it, and then it can go right into the NMR tube with very minimal processing.
We work on structural biology projects related to large molecules like proteins, RNA, lipids, anything like that. And then our second set of projects that we do a lot of are metabolomics. We can look at all kinds of metabolomic fluids. The third set of projects we work on is related to chemistry, basic problems of, is this chemical the right one? Did this get dissolved in my solution? Or is this chemical reaction going the way I expected?
Really the only way that we were able to understand what was happening between this disordered protein and its binding partner was using techniques that are only available to NMR spectroscopists. One of the benefits of using techniques like NMR is the fact that it gives you atomic resolution, particular proteins that are perhaps more dynamic, which couldn't be captured or fully understood with rigid techniques like crystallography.
I came to NIEHS with the idea of solving this RNA structure using NMR. So I came down to the facility, talked to the scientist here. I was able to get training straight away with Eugene. I sat next to him and he took me through all the instrumentation, all the scripts that needed to be written and things like that. And we were able to get sample in the instrument quite quickly as soon as I wanted. And it was very collaborative. Whenever I had sample ready, I came down and it was very, very helpful.
It's sometimes crucial to the stability of the protein sample to be able to just run downstairs, put it in the magnet, and get the data you need within, half an hour.
So the advantage is that the researchers are right here. We can work interactively with them on this worked. Maybe we could manipulate this in a different way. A lot of our projects are long-term projects. We work with people for a long time. It's not we could ship this off to someone kind of experience.
The NMR facility is located off of the F module in the white building that is beneath the first floor. We are in a separate building because the magnetic fields of the NMR are very strong and they're very large machines.
A lot of our research is collaborative, trying to ask questions that haven't been asked before. So maybe looking at a new chemical reaction or looking at molecules that haven't been studied before. And we may need to invent new techniques or think about the best ways that we can manipulate the NMR to study the question that they're interested in.
I think it's also neat that we have the variety of options that we do with the different collaborations between cores. And it is really exciting that we get to study these proteins in the setting of using all the different cores and all of the different resources that are available to us.
The core has invited me to some of the lab meetings, which has been a wonderful collaboration because you can share your data and then also see how the core functions and what other things they're kind of working on, and it gives you a better idea of the experiments that are available.
The biggest changes are these newer, faster experiments have come online. Some experiments, for example, that used to take days, can now be run in hours, or experiments that might have taken a week can be run overnight. There's also been improvements in sensitivity, which allows us to look at smaller and smaller samples.
I can say that it takes a considerable amount of training and knowledge to perform these kinds of experiments. It's such a niche, it's not something you're gonna just pick up.
Working with the staff at the NMR facility was fantastic. The communication was on point. I would email any of the scientists in the NMR facility and they would get back to me within minutes, I would say, and say, bring your sample down, we'll run it for you.
And I really like the fact that you can feel like you're doing something every day, and in general you work with people that are easy to work with, and every day there's sort of something new to look at.
So having lots of resources available to trainees like me gives me the ability to continue to learn and further my career and education before I go into the field.
If you have an idea for a project and you think NMR might be applicable, we're happy to talk to you and let you know how we might be able to work on that project with you.